In the 2024 Paris Olympic Games, Grace Brown won the first ever gold for Australia in the women's individual time trial cycling. Tamarat Tola from Ethiopia set a new Olympic record in men's marathon competition. While watching Olympics is exciting, have you ever wondered, why are these athletes so good? Is it the day-to-day hard work they put in, or something different in their genes that made them talented? While effort is important, it is likely their genetic makeup and inheritance of higher VO2 max that make them better than most of us. Welcome to the genetic basis of stuff and seeds. You have probably heard of VO2 max, which refers to the oxygen uptake attained during maximum exercise intensity. Although several studies in the development of sports genetics have pointed out the possibility of individuals having blessed genetic endowment which contribute to high VO2 max, it was not until 1971 Vassilis Klissouras published a paper named Heritability of Adaptive Variation, and for the first time, the heritability of VO2 max was explained using an experiment and scientific analysis. His study observed the intrapair differences in VO2 max between pairs of monozygous and dizygous twins. Monozygous twins result from a single egg being fertilized by a single sperm that share all their genes. Dizygous twins, on the other hand, result from two eggs being fertilized simultaneously by two different sperms, causing them to share only 50% of their genes. Hence, if VO2 max is a genetically heritable trait, the intrapair difference would be smaller in monozygous pairs. To justify his hypothesis, Klissouras let the twins run on treadmill in progressively increasing intensity until exhaustion and measured their VO2 max. The results showed the intrapair difference tends to be greater between dizygous twins than between monozygous twins. This trend was portrayed more clearly by plotting the pair values against each other on XY coordinates. The data points of monozygous twins tend to be clustered around the line of identity, whereas data points of dizygous twins are more widely scattered. The environmental influence on VO2 max was comparable for monozygous and dizygous twins because of similar prenatal and postnatal environment. The interaction between heredity and the environment did not play a significant role in affecting VO2 max, as none of the subjects were exposed to training stimulus prior to the experiment. It was concluded that heredity accounts almost entirely for the difference in VO2 max. Now you might wonder, which gene really play a role in determining our VO2 max? For many years later, technology was limiting our ability to identify specific genes that lead to variations in individuals' VO2 max. In April 2003, the fully sequenced human genome was completed for the first time. This was done by the well-known Human Genome Project. Shortly after, a paper published on APA identified a DNA transcription factor called hypoxia-inducible factor 1, or HIF1. One of its subunits, HIF1 alpha, is encoded by a gene called HIF1A. A SNP on the HIF1A gene was found to cause variations in VO2 max. SNP stands for single nucleotide polymorphism. It refers to a single base variation in the DNA sequence. Through polymorphism sequencing, the study found that subjects with one or two copies of the T allele at the HIF1A A2500 T locus showed significantly lower baseline VO2 max prior to any aerobic trainings than subjects that are homozygous for A allele. In competitive sports and elite sports, it is rare that athletes do not receive any training. Therefore, sensitivity of VO2 max to training can be an important criterion in selecting athletes. A genome-wide association study in 2013 identified the ACSL1 gene being consistently rated at the top of the list of associated genes. Several other genes were found to also have effects. As gene technology continues to mature, genome can be analyzed on a larger scale. Genomic scans have identified markers on chromosomes 4, 8, 11, and 14 to be linked to VO2 max in untrained state, and markers on chromosomes 1, 2, 4, 6, and 11 to be associated with VO2 max trainability. At this point, you're probably confident in saying VO2 max is indeed dependent on genes. While some people are gifted with genes for higher VO2 max, which help them to achieve more in sports, genotypic advantages do not guarantee success in sports. Like other complex polygenic phenotypes in human, VO2 max variations are influenced by both genetics and environment. This comes down to the long-standing debate of nature versus nurture. Several studies indicated the probability of significant improvements in VO2 max occur in response to high-intensity interval training. However, it is hard to say whether genes or training contribute more to VO2 max variations. In fact, it is more likely both are comparably important and may have interactions such as the ACSL1 gene mentioned earlier, which is responsible for VO2 max sensitivity to training. It seems more accurate to say nature and nurture together determine VO2 max and subsequent potential in sports success. Coming back to the question why are the Olympic athletes in endurance sports so good, we can now say it is a result of both genetic and training factors. Therefore, identification of talent and a well-planned training system together would lift an individual up to stand on the podium of the Olympic stadium.